We are looking forward to an exciting session today sponsored by Signal Solutions that will highlight a non-invasive method for studying sleep, wake, and breathing in rodents. The presentation will focus on methodology, best practices, and use case studies of the piezo sleep system and how this technology can answer questions about animal behavior, phenotyping, and relationships between sleep and disease. Our speaker for today's webinar is Dr. Bruce O'Hara, Professor of Biology at the University of Kentucky and co-founder of Signal Solutions, LLC. He obtained his PhD in human and mammalian genetics from the Johns Hopkins School of Medicine and later spent 12 years at the Stanford University Center for Sleep and Circadian Neurobiology, where he established a research program directed towards an understanding of sleep using higher throughput behavioral and genomic methodologies. Bruce has over 30 years of experience in mouse genetics and mouse behavior and has over 75 publications in areas ranging from the effects of drugs to large-scale genetic screens, including those that utilize the piezoelectric sleep-wake monitoring system commercialized by Signal Solutions.
I thought I'd start off a little bit on the importance of sleep to health and disease. I know most of you out there are sleep researchers probably and don't need to be convinced, or even if you're not sleep researchers, have had personal experience knowing that if you don't get a good night's sleep, you just don't feel right. We don't know exactly why, but we have some increasing ideas why. Also a lot of progress more recently on the interaction of sleep and a wide variety of different diseases, both in effects from the disease on sleep, but also bad sleep on the disease pathogenesis. Then I'll just jump into some of the physiological outputs used to track sleep. Again, most of you are probably familiar with EEG, EMG, the gold standard, if you will, but then the physiological outputs that we use, largely the breathing rhythm and general activity. And then the benefits of our non-invasive sleep system. Again, we use this very sensitive piezoelectric film along the flooring of a cage that gives us this very sensitive output, even down to detailed breathing rhythms and any kind of movement at all. Then how to set up, since we use a piezoelectric film, we call our main acquisition system a piezo sleep. And we'll talk about setting that up, then how the data is collected, which, again, all of these steps we've fully automated in a nice turnkey system to make it easy to use, so we think our system can be used by non-sleep researchers who want to get sleep data on their mouse models or other kinds of experiments. So then I'll discuss a few use cases on how we and others have used this system. All right, just a little background on sleep. Again, most of you probably know sleep is highly conserved certainly across all mammals. It's very similar. The pathways are very similar. The genes that seem to regulate sleep are very similar. The brain areas are all very similar. Obviously, mice and rats are nocturnal, so they sleep more in the day. We sleep mostly at night. They are very polyphasic, but so there are those differences. But in general, the basics of sleep, the basics of the EEG patterns even, are very similar across all mammals. Again, mammals do vary quite a bit in the amount of sleep they get, but all mammals seem to need sleep, so it seems to serve some, obviously critical function. Even lots of work in invertebrates now, including especially in fruit flies. A lot of groups have studied sleep, we know, and even most non-mammals and non-vertebrates that have been looked at also appear to sleep, so it may be even a fairly universal trait, but certainly among mammals, translating from mice and rats to humans is widely done and well justified. And then we can get to some of the general importance of sleep and health. Obviously affects performance and mood, even though it's still unclear really why, although we have a few ideas now. So just a quick summary for those less familiar with the field. We don't know fully why we sleep at all, but we know if you don't get enough, even just going from 7 hours a night, say, if that's your norm, to say 5 hours a night, just that couple hour decrease in sleep, we know, can impact a wide variety of things. Some people, even nobody in the audience, I'm sure, can even become cranky or irritable with sleep loss. There's a cognitive impairment. Almost every cognitive test gets worse. even basic things like reaction time. This kind of points to muscle, but that's really a brain function to the increased reaction time and decreased accuracy. And it's been shown in many, many studies, for example, that driving skills, which involve everything from simple reaction time to more complex cognitive processing, that driving skills drop dramatically with, again, sleep depth. Several nights of 4 or 5 hours can be equivalent to driving legally drunk. One all-nighter equivalent to driving legally drunk. Like drunk driving, there's a wide range of variability across individuals. Some people just get a little bit worse, some people get a lot worse, but it's clearly an impact. A lot of interesting work on sleep and the immune system. Jim Kruger and others for decades has shown these interesting links between cytokines and sleep, and there's more and more evidence that, again, just a modest lack of sleep from 7 hours to 5 hours a night impacts even basic infectious diseases. One study took volunteers and injected rhinovirus in their nose, and people who were well rested with 7 or more hours of sleep a night, generally, although they got the virus gets into the nasal epithelial cells, only 20% go on to a full-blown cold. The more poorly rested people that got, say, 5 hours or less a night, 60% went on to a full-blown cold, 3 times the risk of a cold. Even just 1 hour of sleep impacted the common cold, doubled the risk. That modest decrease in sleep also impacts things like type 2 diabetes, obesity, metabolic syndrome. All of these things go up with, again, just a modest decrease in sleep. Again, most of our work's been focused on mus musculus, the house mouse, common laboratory mouse, and that's, of course, been used as probably the model organism now among mammals in all of biomedical research, and that's been increasingly true in sleep. Of course, rats have been maybe the dominant model for a long time before then in neuroscience and generally and in sleep research, but I think with the advent of better genetics and genomics in mice, the huge number of different inbred strains, the much greater ease of making specific knockouts or other kinds of transgenic mice has really led mice to become, I think, even more commonly utilized to investigate a whole range of things from biochemical and genetic pathways in sleep, anatomical pathways. Of course, a lot of that was done in rats as well, but relationships between sleep and disease. So again, a lot of you are interested in models of human disease, and again, mice have become really the dominant animal models there. So many people have looked at links between Alzheimer's and sleep, such as Holtzman's group at Washington University has shown links between not only does sleep get much worse with Alzheimer's, bad sleep may contribute to Alzheimer's. Behaviors in sleep, there's anything from looking at circadian behaviors interacting with sleep to feeding behaviors. There's more and more work looking at high-fat diets, food restriction, and so on that has wide-ranging impacts on many behaviors, including sleep. And then again, addressing what is the fundamental function of sleep. So I won't spend too much time on this background, but I'd highlight one of my favorite examples was this paper from 3 years ago or so from Make and Nedergaard's group that was done in mice, suggesting that one function of sleep may be to flush the brain. About a year before this paper, they proposed this new idea that the brain has this lymphatic system rather than a lymphatic system. Most organs of the body can exchange fluids with the lymphatic system between the blood supply and the lymph system, but the brain is largely devoid of that because of the blood-brain barrier. There is a little bit of lymphatic tissue around the brain that a group at Duke recently found, but that's most of the brain doesn't have a good exchange with the lymph system. So it uses this lymphatic system, G for glial cells, and all the aquaporins and water pores they have through there that creates this flow from the cerebrospinal fluid through the interstitial spaces, and that volume seems to increase about 60% during non-REM or slow wave sleep. And that increased space allows a 5-fold, that's 500% increase in convective flow during this non-REM sleep. So this may be one of the core functions of sleep, to flush the brain of toxic metabolites, including things we really care about, like she measured A beta peptide. Again, that link with Alzheimer's disease. That is greatly the flushing of that and is greatly improved during sleep. Okay, so now just jumping to some of the physiological outputs used to monitor sleep in mice. We have, of course, the gold standard, again, most of you all are familiar with, EEGs, electroencephalograms, EMG, electromyograms. These are, electrodes in humans just placed on the scalp, but in mice or rats, you have to actually do surgery and put screws into the skull to get closer to the brain. And then other electrodes into the muscles, usually neck muscles in rodents, jaw muscles in people, and that can be used to determine not only sleep and wake, but the two main forms of sleep, REM sleep and non-REM sleep. And most REM sleep is maybe 20-25% of our sleep time, non-REM, or in rodents also coinciding with slow wave sleep, about 75 to 80% of the total sleep time. Other people have tried to use video to distinguish sleep and wake with mixed success, and videos often had a difficulty distinguishing non-REM, distinguishing quiet wake from sleep. And most of the automated systems for video have had difficulty doing that. And so that's been, again, difficult to make as a high throughput system. Other people have used some simple activity measures of IR beam breaking. And again, for very active animals, that works reasonably well in that animals that are highly active will be active during wake and inactive during sleep. But again, I think we'll show that our system is much more sensitive and picks up many more variables. And again, our system relies on these piezoelectric sensors along the floor of the cage so that when the animal's asleep, about the only movement is the chest and abdominal cavities moving in and out. So we get this nice respiratory trace. When the animal's moving, we get these nice big signals. And so we have a very easy way of distinguishing it. This whole idea actually goes back to actually my days in Stanford University and Craig Heller's group, where Dennis Grahn actually first used this to look at rat pups to see can these sensitive films even pick up things as sensitive as breathing rhythms. He was interested in the apneas and stopping of breathing that occurs commonly in developing mammals. Human infants have this at 2, 3, 4 months of age. They'll just stop breathing for 5, 10, 20 seconds. Or in rats and mice, it might be 3 or 4 seconds, or here about a 6-second apnea. He compared, the top one shows a rat pup sitting on a piezo electric surface that has a nice breathing rhythm, stops breathing, and he compared that to an impedance pneumograph, which required a surgical implant, so it's invasive, but just to confirm that yes, indeed, these piezo films pick up breathing and thus could be used to monitor sleep and wake as well. So again, looking at our system, where you can just you can either just have this non-invasive system with a piezoelectric film on the bottom and throw your animal in the cage, versus EEG, EMG, which has this laborious surgery of implanting electrodes, getting a head mount and a head plug on top of the animal. Takes a lot of skill to get good quality. It takes quite a bit of time for recovery. Most people give the animal a week or two to recover from this before their behavior becomes normal. Now, of course, the pros of EEG is that it does differentiate non-REM and REM sleep states. It is the historically accepted gold standard, and it is highly accurate when everything is working right. But even this gold standard, having been in several labs that do EEG, can be highly variable, surgery to surgery, animal to animal, and doesn't always work well. But it is, in the best cases, it is very accurate. But it does involve these highly invasive surgery. It limits the mouse activity because it's wearing this big head plug, typically tethered through cables. There are wireless systems, but they still require a transmitter to be implanted, a head plug, and tends to have more noise than the wired systems. But in either case, that limits mouse activity. Again, very labor intensive and low throughput, not just the surgery, recovery, cabling, but really the time to score EEG. Again, it's surprising maybe after this many decades, there's not a good automated way of doing EEG, EMG, but almost every lab that does mouse and rodent sleep has to score by hand. They typically may use an automated system for initial scoring, but you still have to go back and double check that and have a human look at every epoch to confirm that the automated scoring is accurate. So that's probably the biggest bottleneck in EEG, EMG surgery, and it makes it very expensive and labor intensive. Again, our system, you can just, again, put the mouse in, so no surgery, no head mount, simple to deploy, much higher throughput. We fully automated every step. So again, I think it's lots of labor saving. One con is we don't currently guarantee REM, non-REM discrimination, although we do have software now with a beta test version for this. It's been known since the discovery of REM sleep in the 1950s that breathing becomes much more erratic going from non-REM to REM, so that we do have a marker, and we do see this in our piezo signals pretty clearly. We just haven't fully validated exactly how accurate that is. We think for total sleep and wake, we are as accurate now as EEG, EMG. We published several papers on this, comparing simultaneous EEG, EMG, and lots of other groups have done it too with our system. But again, the REM, non-REM, we're still working on. Actually, Dr. Paul Franken in University of Lausanne, a leading mouse sleep person, is currently looking at this in much greater detail to see exactly how accurately we can distinguish REM and non-REM. But again, I would argue for many studies, sleep and wake can give you lots and lots of good information even without the REM, non-REM distinction. All right, just to give you a quick comparison then, some of our earlier validation studies with our piezo signal. Again, during sleep, we typically get this nice regular signal that's pretty much just the breathing rhythm. Here in the EEG, you can see typical higher amplitude, slower waves that then upon wake become higher frequency, lower amplitude waves. The EMG picks up activity, of course, during wake, kind of matches the piezo. But notice our piezo is the cleanest, easiest to score signal by far. You can obviously see sleep, then wake when the animal starts moving around. This makes the automated scoring as accurate as human scoring, and so again, greatly simplifies our system. So again, to summarize, that mice are very small. They have very rapid breathing, typically 2.5 to 3 hertz in the most commonly utilized inbred strains. So again, this is a pattern during sleep where you get this, again, very regular, consistent pattern. When the animal's awake, much more erratic based on whatever movements. Even if they're in quiet wake, they're always usually moving their head around, sampling the air, and so on. So we can distinguish even the quietest periods of wake from sleep. Here's a case where this is auto-scaled, so this is actually much bigger amplitude than here, which you can see over here. The red is the raw piezo signal. So here's a period of wake, then the mouse goes to sleep, a brief couple-second arousal, and this is quite common. Anybody who's looked at mice in detail, they'll sometimes be in sound sleep, just lift their head up, look around for 2 seconds, right back to sleep. Okay, of course, they can have some much longer bouts, but this is quite typical of mice. The blue is our decision statistic, which looks at several features and variables of the signal. It looks at regularity of the signal using autocorrelation, using the wave envelope, using things related to the power spectrum to see that 2.5 to 3 hertz signal. And those are all summated together to create our decision statistic to tell our system whether the animal is asleep or awake. Negative values reflect wake, positive values reflect sleep. They're more positive because everything's more regular. These are all measures of regularity, basically, that tend to be high in sleep, low in wake. So how does our system actually work? It's the piezo sleep system. We think it's, again, quite easy to set up. The heart of it is the flooring of the cage. We put this piezoelectric film. This is actually a PVDF, polyvinylidene fluoride piezo film, that's then coated in metal, provide electrical outputs on the top and bottom, and then coated again in plastic to protect it from urine, feces, and so on. We then place this piezo film on top of a rubber pad, and then we slide that into our platform. And then we have a cage that's bottomless that sits right on top of this. We then have another cage shield, we call it, that allows you to put bedding on this. But again, even this raw piezo film is well protected. The outputs from the piezo film, again, piezoelectric, piezo for pressure. It gives electrical potential differences when there's any kind of pressure or movement on this film, and then that output from the wires goes through into our custom amplifier and filter, which has been designed to optimize the signal. We've spent 15 years really optimizing the best kind of amplifier and filter for this particular signal, and then it goes into our custom A to D board, essentially, or DAC, for the data acquisition system that you can then plug this directly into, and we've integrated a number of other features in that I'll describe in a moment. So then again, once you put the cage on top, plug everything in to the DAC, and then you can place your mouse in. I didn't show earlier, here's the actual, you can barely see the mouse shield right along here on the bottom, which again contains all the bedding. Some people have worried, does the bedding dampen the signal? And it turns out that when the mouse is right on top of the bedding, that signal goes right through to the piezo film, right through the cage shield, and we see almost no loss of signal. And people have used all different kinds of bedding, has not been a problem, and we get good signals with pretty much any kind of bedding. If people want to use nestlets or little igloos and so on. Here's again a setup of 4 cages, each plugged in. This is one of our 8-channel DACs, or 8-channel, we call them squid boxes now because all the cables kind of look like a squid a little bit. And you can plug these in. We have boxes anywhere from 8 to 80 channels. All right, so then collecting data, once you've plugged it all in and have your mouse in there and you want to collect data, you can, the screen will pop up automatically once you plugged it in and you select the DAC. Most people just have one DAC, so you just click OK on that. It'll know that whether you have an 8-channel DAC or an 80-channel DAC, this one's an 8-channel, and you can, however many mice you have, you can select that number. You can also record from an empty cage if you wish. You then put in whatever your light-dark cycle is, because we have automated output that will help you analyze that data. Most people want to know how much their mouse is sleeping in the day versus the night period. So here's a pretty typical case of lights come on at 6 a.m. They go off at 6 p.m. or 1800 hours. 12-12 cycles, I think, work much better for sleep studies than 14-10. Talk about that later. And then you enter the mouse IDs. Here we have male 1, 2, 3, 4, just 4 mice. You click OK. Then a summary sheet comes up, making sure you have everything entered. OK. And you can just then click OK and start the recording. So here's what it would look like. You just start the recording here, and the upper panel gives you the raw piezo signal you're getting at the time. Power spectrum analysis from 0 to 15 hertz. We typically have the filtering that cuts off a lot at the higher frequencies, which is why this drops off so dramatically. And so the real action is really from 0 to 8 hertz information. We have a decision statistic that's based on all those quantitative variables I described, and breath rate intervals. Okay, and so then you can see this. Start up the video again. It's going on. Here is a period of, you can see, of wake and then back into sleep. And as we progress further in this, you'll see as we get into a more clear-cut period of sleep, it auto adjusts so you can see the signal better, and you can see that this is now a clear-cut sleep and breathing signal of about 2.5 hertz. So on the power spectrum based around similar to FFT, we get a nice peak at 2.5 hertz, typically a peak at the harmonic at 5 and 7.5, but that's a very clear-cut signal that the animal is in sleep. Again, we have many other variables besides just the power spectrum that relate to our decision statistic that takes into account regularity of that signal. So again, you can still see the nice peaks at 2.5 hertz. The animal's asleep. And this is now channel 1 or cage 1. You can now look at channel 2, channel 3, or you can go over to the signal panel, look at all 4 of your cages at once. So here you can see that all 4 animals now are asleep. We have a green bar that lights up when the animal's asleep based on a quicker algorithm we have. And note that the amplitude does vary a bit because sometimes mice will sleep really flat with their belly against the bedding, and other times they'll be curled up with their head curled between their legs more on their side and typically get lower amplitude. So we don't use amplitude as a way to determine sleep, but the regularity of the signal, the frequency of that signal at the 2.5 to 3 hertz, and things like that. So again, we've been working on this for, again, over 10 years, and we've really optimized these algorithms to make sure we score sleep accurately with all of these. We've done that, again, with simultaneous EEG, EMG validation. Next, you can look at a sleep summary after a few hours or a day or 2 days. People typically, for sleep studies on these, might do anywhere from 3 days to a week or even many months if you want more circadian type variables or longer term sleep studies. Again, since our system's automatically scored, no limit to doing 60 days straight of recording. You have to change the cages once a week like normal, but that's about it. So here's, again, after that, the sleep summary, the decision statistic. You can look at the data again, make sure you entered everything, or maybe you have auxiliary data if you are doing simultaneous EEG, EMG, or if you want to have some sleep disruption automated method that's giving you additional data, or applying changes in air-gas mixture people have done with our system. So all sorts of things you can have under this auxiliary data. Okay, and then just a little more details on our data acquisition system. We've, again, customized our own DACs. These we call squid boxes, and or calamari boxes for our smallest 8-channel one. Here's an 8-channel box, can handle 8 cages. Here's a 64-channel one that can handle 64 cages. We have our giant squid size goes up to 80 channels, which we've had places like the Jackson Laboratory burning 80 mice a week for 5 years, or you could even have 2 of those. And again, this allows not only for the analog to digital conversion of the signal, but has buttons you can push to mark for data marking if you're doing something at a given time, changing gas mixtures, sleep disruption. You can have other digital or analog inputs or outputs from this system, so it integrates all these capabilities. So again, piezo sleep is our data acquisition system. Then all that output can be quickly analyzed and give a lot of the variables you might like through our sleep stats analysis software. And you can get your percent sleep times in the light period, the dark period, over 24 hours. If you're doing multiple days, you can average all those days together. You can throw out the first day if it's disrupted and do whatever you want. The other major variable you can get is bout length distribution, that really measures sleep consolidation, fragmentation of sleep. That's important. It's not just the amount of sleep we get, right? It's the quality of sleep, which is also important, and consolidation is one measure of that. Other variables we can get, we also have general activity measures, so you can get how active your mouse is throughout the day, and the activity onset, which in most of our light-dark studies, it'll be right around dark onset. But then also we have measures of peak activity during that 12-hour night. It automatically generates these CSV files, so you can now look at in great detail at hourly sleep values for bout length or anything else you like to do in Excel or other programs. So the initial output from the piezo sleep will give you this file that's our feedback file for all the features that are summated, and from there you can just click on that, and it will analyze now all this data and give you this menu on the left where you can now look at various aspects of percent sleep, bout length, and so on. So here we'll pick as the individual, mouse number 1, and look at it. And now here's the decision statistic over time. So here was like a 4-day recording, right? 120 hours would be 5 days, but started just before dark onset, say around 4 in the afternoon. That first couple hours can actually be interesting to analyze in its own right, because mice respond to that novel environment differently, but a lot of people just throw that out if they want kind of a baseline sleep, maybe starting on the second dark onset and analyze these 3 days, or maybe at this light onset and look at these 3 days to average. Or you can run it for a week, 2 weeks, whatever you want. And again, the decision tends to be negative during wake and positive during sleep. So you can see this is dark onset, light onset, also shown by the bars here, dark and light for 4 successive days. You can see a lot of wake during the night, a lot of sleep there. But even during, mice are very polyphasic, so you'll also see a fair bit of sleep here and a fair bit of wake here. Average, say, CFD7 black 6 mouse might sleep 28% of the night and 63% of the day is pretty typical. So this gives you kind of an initial look at your data. You can then blow this up if you want to look at it in more detail. So often I say take these like 7 hours, and you can then superimpose from this menu the red actual raw piezo signal. And again, you can see that when there's lots of wake, there's lots of these big amplitude signals. When there's lots of sleep, there's lots of the small regular signal. If you actually blow this up, you'd see some bigger bouts of very short periods of the other. But we can do that now. Actually, we can blow it up even further and pick a period of sleep, and now you can start to see the regular pattern in that sleep. Maybe blow it up one more time and take a look, and now you can see that the classic 2.5 to 3 hertz pattern. This is actually 0.0002 hours, which is about a second. From this tick mark to this, you can redo it in seconds if you prefer. And this is, again, a very clear-cut case of regular breathing, regular sleep, and so on. So let's see, let's start that up again. Okay. Then you can, again, go back to the menu and look at other automatic outputs we provide. So another nice, easy one to look at is, let's say, here's your 4 mice. You can look at how much they slept in the light period, how much they slept in the dark period for each of your animals. And again, that's a pretty typical patterns for rodents. Another one I like to look at is this wake graph. And here we do percent wake rather than sleep because it's nice. You can see that typical activity burst most mice will have, pretty much all the common inbred strains, and unless you have something really weird going on with your mouse, which hopefully you know about or you can look at in detail if you do have something weird, but most mice will have this great increase in wake right at dark onset for anywhere from 1 to 3 or 4 hours. And then mice do, again, sleep a fair bit in the night too, especially, say, black 6 mice. 3/4 in the night will have another, a little bit bigger sleep out period. So this is a very typical pattern. But this is a nice 4-day look to see if everything's working well. This nice increase each dark period for 4 successive days. We have other variables looking at the dark weight ratio of, so give you an idea of sort of diurnal amplitude, kind of for a circadian variable, and so on. All right, then you can go back here and again pick our individual mouse now to look at bout length. So you can click back on that menu and now look at bout length. In this case, we've set the default variable for Windows at 30 seconds. So the shortest bouts are 30 seconds. And with that, you're going to be ignoring the 2-second, 5-second, 10-second things. You would need 16 seconds of wake or 16 seconds of sleep to interrupt that bout. And using that kind of window, you'll typically get, say, a peak of about 1,000 seconds or 17 minutes for bouts at night, and maybe twice that long during day on average. Bouts in the day are a little bit longer than night. This shape of this will look about the same. If you pick shorter, for some kinds of studies, you might want to put this at 4 seconds or 5 seconds and look at everyone. Others, this is a good default. So I'll show that in a little bit, how you can reset that. Next, you can now look at that decision statistic. Here's a case of a nice bimodal one. This is presumably a mouse that was probably highly active when it was awake and then sleeping, so you don't see that. Remember, some of the other ones earlier were kind of trimodal because distinguishing quiet, wake, and sleep. This was a nice easy one. It's very clear cut. Next, you can, again, we talked about it automatically exports all your data to CSV files, so you can pick exactly what you'd like to export, whether it's, in this case, hourly values, 60 minutes. You can, again, set that whatever you want, and you can now export that. In this case, you can hit mean sleep bouts. It computes all those mean sleep bouts now, and it will give you the output into a data file, which you can then open in Excel. And now from Excel, you can have your data on your 4 male mice in this case. Let's say these were let's say you want to graph these and look at them individually. You can do a bar graph, look at each one individually, or perhaps this was 4 mice all part of one grouping, and you want to get averages for those 4. You can then just average them and then very quickly get data on what your average for that group was for your sleep consolidation, sleep fragmentation, average bout lengths. Okay, and of course you can do then whatever other statistics you like to do in Excel and things like that. All right, here I mentioned you can change this variable. Here the default was 30. You can change it, say, to 5 seconds. Now you can pick up all those little 3-second arousals and 3-second sleep outs again. I find for most comparisons it comes out about the same, but for sometimes you really do want to see those short ones, so you can, again, redo the analysis under those conditions and just reset things. You can then save those changes or go back to the previous menu, or just, in this case, we'll just quit the file, exit the program, and move on. Okay. So again, I think we've automated the acquisition, we've automated the analysis, and now again, what are some of the things we can do with it? I'm sure you all maybe have your favorite ideas, but I'll share a couple things we've done. We think it's made much more doable and easier to do large-scale genetic studies. So we've done QTL mapping studies and BXD recombinant inbred mice. Our system was used by the Oak Ridge National Labs in 2008, 9, and 10 to study all the pre-collaborative cross mice. That paper was published in Genome Research. We've looked at a lot of individual mutants and knockouts, testing of drugs on sleep. There might be a lot of in drug development and so on, assessing sleep quality in mouse models of disease. So again, many groups have looked at sleep effects and mentioned Alzheimer's models. We've done some of that ourselves, or after traumatic brain injury. We've done a lot of work on sleep with Johnny Lifshitz, Rachel Rowe, and others who were at Kentucky and now at Arizona. And our biggest project so far has been this large collaborative effort with the Jackson Laboratory. I'll describe some of that data in a minute. And again, we think we've allowed this to be done in a much more cost-effective way, no particular expertise needed. The Jackson project was actually part of an even larger international effort to ultimately try to do deep phenotyping on all 22,000 protein coding genes with single gene knockouts, and the JAX being, of course, one of the major centers within the United States for this. But all data is shared throughout internationally on this. And the COMP project, the knockout mouse phenotyping project at JAX, over the past 5 years, we've looked at over 10,000 mice, which is a lot to do for sleep, which represent about 300 different mouse lines, males and females, and we had one of the highest hit rates of any phenotype, and I think that's because sleep is one of the most defended behaviors an animal has. So black 6 mice, which is the background here, males will sleep about 43% of a day, females about 41% of the day, very consistent, much more consistent than most behavioral tests, much more consistent, we think, than most physiological tests. So we would argue sleep is one of the cleanest, simplest behaviors you can add if you have an interesting mouse model. The genes selected in this system were unbiased, not necessarily ones we thought would be important for sleep. So this was 300. You could think of almost random genes, many of which we don't know a lot about. That was one of the ideas of this project. We could look at the dark matter of the genome of the 22,000 protein coding genes. So to understand purpose, function, insights, and pathways that would affect sleep and a wide variety of other physiological traits. As mentioned, we also pick up breath variables. And it turned out in some cases things that were all lung assessment and issues with breathing and lungs also correlated with our breath rates during sleep, so we provided some information on that as well. Again, we think this provided a nice unbiased approach to find genes. Again, of these 50, 5 of them, of those 300, were some of the biggest changes in sleep ever found for any knockout, even when people picked their favorite sleep-related genes. This information is all available at mousephenotype.org. All information throughout the COMP2 and the IMPC is made publicly available, so any of you all can look at that in detail if you wish. Just a second example I thought I'd show for a look, those interested in drugs or drug development. Here we have another collaboration from the College of Medicine at the University of Kentucky. They actually had this Chinese herb that really not much science known about it, but a lot of anecdotal reports from users that it improves sleep consolidation at night. But it's not a somnogenic. People could take it in the day. It might affect their quality of sleep later. But really very little information on it. So initially we got our basic total sleep times, not much different. Not shown here, but the day-night sleep times also not much different. But then we wanted to look at, did it affect fragmentation or consolidation? So we took those, in this case we start with hourly values. In this case we started at 9 minutes after an hour, so the hours are all consistent with that. And we take our output of that hourly data and we can export that to Excel. We can plot that automatically, and now we can look at, let's say, maybe I'll go back. So there were 4 doses of 0, 5, 25, and 50 mg. Just to keep it cleaner, I'll show just the 0 and 50 mg, which had the biggest effect. And you can see in the light period, here's the those that got the herb, and here's those who did not. This was given by oral gavage. And much better, much longer bouts. Again, more of these around 25, 30-minute sleep bouts, many more in the dose group than the undosed group, which was highly significant. Also true in the dark period to a lesser extent. You can see, again, more of the orange at these longer bouts than the blue kind of ends here. At the 0 dose, again, bouts tend to be shorter in the dark. So in both dark, but especially in the light period, the time when mice really normally sleep, it did seem to match these anecdotal reports, which is actually rare for, I think, in my experience. Also, this group's done a little bit of work with different effects of dosing at different times of day. This graph is from doses given the daytime, but other times of day cause maybe some interesting differences as well. But in any case, it's another promising case where could be a new drug to help improve sleep consolidation. One more case that I just showed briefly that comes from collaboration with Ashley Seifert's lab, again at the University of Kentucky, who studies these African spiny mice. Here's the regular mus musculus, house mouse, common laboratory mouse, and the African spiny mouse is Acomis carinus, sometimes called the Egyptian Cairo mouse. It's actually closer to gerbils than mice, so it's actually quite evolutionarily far removed from mus musculus, probably separated 15, 20 million years ago. In any case, these mice are interesting models for a variety of other diseases that could be an interesting model. But the most interesting thing we found immediately was they sleep with their eyes open all the time. You can see here is a sleeping mouse. They might have an eye half open or partially open, or other times you'll see that other animals, horses, cows, even people can have brief periods of sleep, maybe with eyes partly open, but to my knowledge, there's never been a case of sleeping with eyes totally open. I'll show that on the next slide. Here's a case of a sleeping. So here's a very clear-cut sleep signal in our piezo, very clear-cut in the EEG. This mouse is sleeping, and its eyes, you can see, are totally open during this sleep period. Over 20, we've looked at in video, EEG, EMG, you can see the cabling and piezo. And now you can see the animal gets up and moves around. We get the nice bigger piezo signal. So again, just another interesting study. It's also another species. These spiny mouse are about twice as big as mus musculus. We've also done some studies on rats as well. So that's, again, under future applications. We can do other species. We've even done a few lemurs with the Duke Lemur Center down there, both mouse lemurs and dwarf lemurs. So again, the rat actually being bigger gives it actually even much easier to read signal. In fact, they're so big that when they're lying flat, we can even pick up not only the breathing rhythm, but the actual heart rate, which is usually about 2 to 3 times faster than the breathing rhythm. That's ongoing. I mentioned the REM, non-REM discrimination ongoing. Breathing definitely becomes more erratic with REM, so we know we can distinguish it to some accuracy. We just don't know exactly how accurate yet. Other behaviors, we have activity we can already do. You can just summate all the amplitudes, and we already have good activity measures. We give that additional software for anybody who wants it. Also, there appear to be characteristic patterns during periods of seizure, especially active grand mal type, shaking type seizures. Also cataplexy, cessation of movement, loss of muscle tone in narcolepsy. We can track other breathing variables besides just rate, such as sleep apnea I showed way back for the rat pups. We think this could be useful for general health monitoring. I forgot to mention that you can view your data from anywhere. So you could monitor your mice from a distance. Any computer that's hooked up to your computer in the animal room, you can see how they're sleeping, how they're waking, and other variables that may reflect their general health status. We've developed some systems to disrupt sleep and or work with existing sleep disruption systems, or work with cages that control gases of CO2, O2, and so on. Okay, so just for a quick summary then, we think we have this very simple to use, non-invasive system to monitor sleep, wake, breathing variables, and probably some other variables we're working on now. And again, no surgery, better for the mouse, simpler to deploy, it's higher throughput, and less expensive. Just some quick acknowledgments. There's probably, I could put several dozen more people on this slide, but I'd like to at least thank our core Signal Solutions group, my co-founder, Professor of Engineering, Kevin Donahue, more recent addition owner, Mike Lehman, both engineer and business experience, Anuj, biomedical engineer, Rebecca Bernat, our lead sleep and general biomedical research experts. My own laboratory of graduate students, PhD students that have contributed a lot, Sridhar Sundaram, another lab on campus in biomedical engineering, and his students have contributed a lot. The Jackson Laboratory, the major project, has been very successful, we think. Rodent Behavior Core that I happen to direct as well. It's been helpful. Funding from Department of Defense, National Institute of Health, our Kentucky Cabinet of Economic Development through the Kentucky Science and Technology Corporation, and so on. So those are people we'd like to thank.
All right, so Bruce, with that in mind, let's get on to our first question. Several people, including Heather, have asked if it's possible to measure sleep-wake breathing using a system like yours in group-housed rodents.
Okay, yeah, that's something we've been interested too, and we're still evaluating that. In general, mice tend to sleep together and wake together, so you can get certainly a general accurate outline of when your grouped animals are sleeping and when they're awake. Obviously, if only one animal is awake and the other is sleeping, that does tend to disrupt the signal, so we can't always pull that out, but we're working on algorithms that can actually separate that kind of information, so that's, again, in process. So we think we could get some crude data of group sleep, group wake. Getting detailed individual animals within a group is in progress.
Okay. You mentioned this, but would you be able to elaborate on how the piezo sleep system distinguishes between inactivity, so quiet wake versus sleep?
Yeah, so I think I mentioned it, unlike other video or IR beam breaking that can't really distinguish quiet, wake, and sleep, our system does, because when the animal's on typically in a corner, it's almost always moving its head back and forth, so the signal we will pick up the breathing as well, but it'll be much more erratic, and with those head movements and paw movements, so that mice are almost never completely still when they're not asleep. So we think we can very accurately, we've compared, again, looking at EEG, EMG, it probably is where our biggest error is, like especially REM, where breathing becomes more erratic, and quiet wake is probably the toughest distinction we have, but we've gotten, again, comparing to EEG, EMG, we've gotten, we think, pretty good on that as well.
Okay, excellent. John has asked if your system is able to integrate with sleep deprivation chambers.
Yes, yeah. In fact, we've specifically, I'm not allowed to say other brands, we've specifically worked with people using like the Lafayette bar suite method, so a couple different groups already. We've worked with them. Other ones we think we can work with as well, and customize our piezo films for pretty much any kind of chamber. And then we have our own under development that uses a novel mechanism of sleep disruption that's more variable, and we think it really keeps the mouse even more awake. That's down the road.
Okay. Quite a few people have asked about vibration from things like cage racks, and can you talk a bit about, do vibrations in the room interfere with the breathing signal and the functionality of the system, and is there, how does one go about addressing that?
Yeah, that's been a big concern of ours over the years. We think the filtering and amplification we do really accentuates the key signal we need to distinguish sleep-wake. That being said, yeah, some rooms are a bit noisier than other rooms, so it is good to have as quiet a room as possible so there's not too much vibration. Usually, again, even if when people walk in a room, to check on mice, that's only usually a matter of minutes out of a 24-hour day. So even if it's just disrupting then, over a 24-hour period, we find the data doesn't usually be impacted much for most sleep variables. Or if it's really a severe interruption, we can even eliminate that period of interruption. So usually cages aren't vibrating all day long, right? But although we have had cases like of trucks going by and things like that, and that so far has not disrupted our signal scoring. We can sometimes see that vibration at higher frequencies, but so far our sleep-wake is robust enough that we can always score the sleep-wake accurately, even with trucks driving by a facility.
Excellent. Good. Can data be monitored remotely while an experiment is ongoing? So I think, in other words, does somebody need to be in the lab operating this, and obviously one doesn't want to disrupt the animal, so how would one go about doing that?
Yeah, so as long as the animal rooms have internet. Some of the older animal rooms don't, but you can always get a wire in there if you really push them, or a wireless system if you really push them, in my experience. So as long as there's internet capability in the animal facility, then you can monitor this from anywhere. So it is a nice way of even just checking on animals. That's why I think it could have a long-term usefulness as generalized health monitoring, but also for your particular sleep-wake data and breathing data. All the screens I showed you can access from any computer anywhere, as long as your animal room computer has internet access.